All right, hi everyone. We're going to go ahead and get this session started. Before we start, I have some housekeeping notes I want to say to you guys. So please silence your cell phones. Photography of ABRF meeting sessions is permitted, subject to the speaker's request not to capture pre-publication data. Recordings of the meeting sessions will be available on the website following the annual meeting. ABRF's social media policy is that everything can be shared. If a speaker asks that it not be shared, please respect the speaker and do not post on social media. Hashtag for the event is hashtag ABRF 2025. Unless noted by the speaker, questions are to be held by the end of the session, but when you guys ask questions, please use the microphones on the ends here. And you can download the ABRF meeting app to view the full schedule, access lightning talk abstracts, connect with other attendees and sponsors, play the ABRF game, and create your personalized meeting schedule. You'll find extra content, including hours of the exhibit hall and tech showcases to thank your sponsors. And all attendees will receive an overall meeting evaluation survey following the meeting to help improve the ABRF meeting experience. So this session is going to be a panel discussion session, so we're hoping that we're going to get a lot of discussion at the end of it. But we're going to start with a vendor talk from Amos at Meteor Biotech.
Thank you for the introduction, and I'm very honored to, we're very honored to sponsor this session, and also very honored to speak in front of all the people who are interested in the area of spatial omics, the field, I think, which is shifting the modern biology area. So the technology that I'm going to introduce you about today is called SLAX technology. It's a short for spatially resolved laser activated cell sorting. It's a proprietary technology that we developed in a university in Korea, and we named it after FAX technology, but instead of it being fluorescence activated, we use laser to physically isolate, sort the cells, retaining the spatial information so that you can connect that good spatial information, pathological information to the cells, components like DNA, RNA, or protein, so that you could have that targeted deep dive into these cells that you want to analyze. I'm going to open up with how the spatial information is important. So we have a universe of biomass. If you look into the 2D spatial landscape of these tissues, you have the proximity of the cells. You also have the composition of different cells and morphology and tumor or cellular tissue structure inside these tissues. So it's all important. These are all important spatial features, as there are some connections to the pathologically important features that have been the basis of modern pathology and modern medicine. And we wanted to, our motto of the company is what you see is what you get. So you have the what you see part, which could be the spatial assays that are out there, such as H&E staining, multiplex immunofluorescence staining, FISH staining, labeling-based omics staining technologies, and other imaging technologies. And you have the what you get part, which could be the DNA sequencing, RNA sequencing, or proteomics using mass spec, or metabolomics using, or lipidomics using mass spec. So we wanted to have a bridging technology that you could mix and match different spatial assays to omics assays, so that you have that freedom of analyzing your target from tissue while retaining your spatial or pathological information. So SLAX technology uses infrared pulse laser, which is very less detrimental to the compared to UV ultraviolet laser. It's almost pure thermal energy, and we have this very thin glass-like layer which has a similar or same physical property as the normal glass slide would have. So if you can prepare anything on a glass slide, you could also prepare in our layer. And this layer vaporizes from solid state to gaseous state and it is exposed to infrared pulse laser, so that the cells that are prepared on top of it will be physically punched out to 96-well format PCR tubes, so that you could perform any biotechnology that you can do with PCR tubes. So, oh, sorry. Oh. So this is a real-time speed of isolating parts of the tissue. In this case was HER2-positive breast cancer tissue. A pathologist marked off where he was interested in and previously, and the machine would find that place and then sort these cells in PCR tubes so that we could perform whole genome sequencing. Also, the core, the principle behind this workflow is that if you have any kind of tissue, the biospecimen that could be prepared on top of that slide, you could image them and look for where you want to isolate, and then we can target that cell, and then you can have the deepest information that you can get from these cells using whole genome sequencing, whole transcriptome sequencing, or proteomics using mass spec, or any other technologies that you can do with PCR tubes. So after these cells are isolated, we can get the DNA, RNA, and protein information, and then map those omics information back to the spatial information so that you would have that spatial network of different molecular features inside these tissues. A little bit slow, sorry. So if you could do a serial section, consecutive sections, this was a tumor mass from a triple-negative breast cancer tissue patient, triple-negative breast cancer patient, and we were able to take out the rarest subclones, genomic abnormality, so that this patient would have her a bespoke panel for monitoring her relapse and the tumor. And we can also do this in bone marrow smear. This case was a leukemia patient's bone marrow smear. And we were able to pick out that single leukemic cell and then define their genomic abnormality so that this patient would be followed up with his own genomic profiles and monitor their MRD. So we would like to say that our technology is positioned as a uniquely focused on finding the target of the tissue, whereas the different spatial omics technologies are very good at exploring the tumor overall landscape of the tissues. So we have publications in this matter, a review paper where we claim that there are technologies, spatial omics technologies that are more suited for broad overview analysis, such as fish-based, labeling-based technologies, whereas our technology is more suited for targeting that cell and then getting a deep dive into that cell. So we had this demonstration in 2022 published in Nature Communications, where we immediately found a marker of triple-negative breast cancer patients who had received neoadjuvant chemotherapy and had a chemo resistance. So we had to perform surgery. We hypothesized that these would contain chemo-resistant breast cancer stem cell population and wanted to characterize them compared to just cancer cells. And we were able to find RNA mutations in GPX4, and we immediately found that a patient group, from 109 patient group, we were able to see that this marker had a good differentiating point between of survival probability of the patients when divided with the patient group who had this RNA mutation and the patient who didn't have this RNA mutation. So we're taking this further to see if this inhibitor of this RNA mutation could aid chemotherapy for triple-negative breast cancer patients so that they can be used as a companion therapeutics. So as soon as we published that, we were acknowledged by Nature Reviews Cancer as a tools of the trade that makes scientific discoveries possible, and we were able to convert this technology into a commercialized version. And this was in another case that we isolated osteoblasts from a mouse femur, and to see if drug candidates were actually affecting the osteoblasts, reactivating or inactivating, so that we could see if these candidates are suitable for a cure for osteoporosis. So this is a resolution of our technology. We are able to isolate single E. coli from, if you can see in A. We also have cases where we isolate a single chromosome in Giemsa bending, and if you can see, as you saw in the video, we are also able to isolate single cell, fibrous regions, rigid regions like femur, or F, also a very useful tool when you're looking for very rare cells that can be lost in normal cell sorting applications. So the machine on the left was the machine I built as a student when I was graduate school, and the machine on the right is a manufacturable version that we have in Meteor right now. So we've tried this with IHC, multiplex immunofluorescence staining, fish stain samples, and spatial transcriptomic samples like Xenium or CosMx, which are non-destructive to tissues. We could perform these omics technologies and then move on to SLACs to isolate Xenium-defined cells, for example, and perform mass spec. So you can have that good multi-omic approach in understanding the biology behind the tumors. We also have applied, we have a publication in Nature Biomedical Engineering where we define this image feature using AI and then look for the good features. So we're looking for different indications, disease indications. We have 3 sites in Korea and currently 2 sites in the US and 5 more to come in late June. So yeah, this is the team member that we started. We have 10 years of experience of developing this technology. Yeah, so I wanted to conclude this presentation by saying that we want to develop this technology as a bridging tool for real-world applications from all these atlasing projects. Thank you.
Thank you, Amos. That was nice. Okay, so now we're going to start our session about our navigating spatial different technologies. The reason we wanted to do this session is because we know there's a bunch of spatial technologies that have been coming on pretty quickly in the industry, and they all have their different strengths and weaknesses. So we want to go through them so you know which experiments are going to work well on which devices. So I'm going to be your Bruker expert in today's panel. So I'm going to be focusing on the GeoMx and the CosMx for my presentation. So we're going to look at the GeoMx technology, just kind of give an overview of it. And then we're going to do some experiment strengths. We're going to look at the CosMx technology, followed by experiment strengths of that. And then we're going to compare the 2 platforms at the end. So let's look at the GeoMx digital spatial profiler. This is Bruker's sequencing count-based system. So this is their technology in a nutshell. You do your sample prep, and during the prep, we're going to be adding these probes that connect to our mRNA. And at the end of those probes, we have a photo-cleavable probe-specific barcode. So we'll get all those probes on there. We'll add some fluorescent morph markers, and we'll run it through our system. Once we get it through, we're going to have a nice picture of our tissue, and from there we can select the regions that we think that we're going to see our interesting stuff. After that, we're going to do a library prep, and then we are going to send it off for sequencing until we get our counts back. So the GeoMx really focuses on ROIs and regional sections. It's not a cellular, subcellular resolution. It's more looking at regional profiles. So it does get down to like a 50-cell resolution, but that's really as small as we're wanting to get it. So the reason why we can only do that is because the GeoMx has a ton of these itty-bitty mirrors in them. And when we tell it where our region of interest is, it'll position those mirrors so that a UV light hits that spot and releases the probes into solution. A pipette, it literally comes, collects those probes, put them in a well, and it does that for every ROI that we pick. After then, we'll take that, sequence it, and then we have our mRNA parts that are connected to our spatially resolved cell tissue. So the strength of the GeoMx is anything that's really not cellular or subcellular based. So if you're looking at your heterogeneity between your regional expression patterns, if you're looking at different biological compartments, or if you're looking at some of those microenvironments on each side of a tumor, it's really good at picking up areas that go away from your tumor, so you have your different contour patterns, and you can see exactly where some of those cells are changing their properties. So, yep, that's the GeoMx for you. And so now we're going to switch over to the CosMx. And the CosMx is Bruker's imaging-based program or machine, and it goes down to a cellular and subcellular resolution. And the way that it can do that is by this image and bleaching strategy they have. So when we're binding the CosMx probes to our tissue, there's a primary probe that binds to our transcript of interest, followed by a branch secondary probe. And that probe has a sequence of fluorophores that will connect to it. And after 16 rounds, we're going to get a signature that corresponds to the transcript that it was connected to. So this allows us to get inside the cell and grab all those transcripts. So this is kind of what it looks like as the CosMx is running. We're going to be putting 16 cycles of fluorophores on there. So we're going to be putting a red, a green, a yellow, and a blue. And we're going to see where that sticks. And then we're going to take a picture of it, and then we're going to photocleave everything off, and we're going to do it again. And we're going to do it 16 times. So by the end of it, you have this nice little color-coded barcode that corresponds to the transcripts that we're looking at. So some of the strengths of this technology is that you can really look at the different cells and the different characterization that's happening within the same cells in your tissue. You can see why certain immune cells are acting a certain way in this environment versus maybe an environment in a little more healthy region versus tumor. Cell-to-cell interaction is very good with the CosMx. You can look at the physical differences between cells. You can see what they're expressing and how the cell beside it is responding to it, which is very good for any disease or therapeutic options that we want to come with. And then obviously spatial biomarkers. Since we're getting single-cell resolution, you're able to pretty much pick where anything is going to go into your tissue. Okay, so that was an overview, and I know mine's super quick, but I just kind of wanted to give an overview of what these two Bruker technologies do. So, both CosMx and GeoMx only have mouse and human panels. I think the GeoMx actually does have a canine panel now, but they specialize in mouse and human panels for sure. The GeoMx is not single-cell resolution, it's more regional. You want to know your region of interest, and you kind of want to know what you're looking for. Versus the CosMx, which is cellular, subcellular, because you're going to get a lot more information from it. The GeoMx does do over 18,000 genes profiled in their whole transcriptome atlas, and CosMx will do. They have their new 6K panel out for their human and their 1K for mouse. They also have a CosMx whole transcriptome panel in the works, but we're not sure when that's going to come out. The GeoMx can profile quite a few proteins. They've made it so that we can profile our proteins and do a sequencing-based count so we can see the expression levels in our tissues. And finally, the capture area, which is a big thing here. So the way that the Bruker technologies are priced is by slide or capture area. So the CosMx has a little smaller capture area, but you're still going to get that single cell resolution. Whereas the GeoMx has a larger capture area, but you're only going to get information in the areas that you select in your tissue. So that's a big point that you're gonna want to think about if you're looking at the Bruker technologies. Because yeah, as many samples as you can fit in that capture area, the more cost effective it's going to be. And then I just wanted to acknowledge the University of Nebraska Medical Center and the Office of Vice Chancellor for Research and my director, Sean Cross.
Hi, so I'm Silas Maniatis from New York Genome Center, where we are in the midst of setting up a new spatial platform that is service and production scale oriented. And this is building off our long track record of this type of activity in the single cell environment. If I'm eager to interact with anybody who has interest in developing a spatial project with us, so please come chat with me after the meeting if you're looking for a very collaborative service provider. Right, so I thought that rather than try to go through individual technologies and tell you this or that about them, I would show you some case studies on how we go about thinking, about what technologies we need to answer the questions at hand. There we go. All right. So first, I just want to acknowledge an amazing set of collaborators I have. Hamali, Rich, and Joachim are some of the best colleagues and mentors one can have, so thank you to them and to my team. The 4 institutes at the top that sort of drove all this work, just phenomenal places. Of course, all of our funding sources, but most importantly, the patients who donated the samples used in our work. So thank you to all of them. So in the first thing we're going to look at, we started using spatial technologies very early on to explore the molecular underpinnings of ALS, right? So ALS is a terrible disease. It's invariably fatal. Typically from the time that you become symptomatic to the time of death is very short. And we don't know a lot about it. One of the main difficulties in studying it is that only 10% of cases have a clear heritable history in their family. And of the genes that we know are related to ALS, most of them are ubiquitously expressed, yet it is motor neurons that are specifically lost. And so there's a lot of questions there that make pretty much anything other than a spatial approach not very helpful. So we know that multiple cell types contribute to the disease, and so we need technology that's going to be able to allow us to look at all of them. We know that there's very specific areas of the tissue where motor neurons reside, and I'll show you that in a moment. And so we know that bulk approaches aren't going to be great for this either. And at a sort of organismal scale, we don't fully understand where in the sort of rostrocaudal axis ALS is going to kick off, and in what cell types, and what the ordering of these events is. So in this image here, I'm showing you in green motor neurons, and then in red neurons generally, and then in blue is DAPI. So you can see that the number of cells that were actually the most important or most relevant in this disease is quite small. And so trying to understand what's going on with them in a bulk assay is just hopeless. And in fact, when we look at a murine model, we can, at P90, see a couple dozen genes that are differentially expressed. Before that, we know that there's already electrophysiological problems and whatnot, so we expect there's molecular problems. And so we're not seeing them in bulk. Complicating things, a motor neuron can be a meter long, and so trying to isolate a single large motor neuron in a swamp of tiny little cells many of whom are, making myelin and, causing all sorts of debris in a single cell prep. It just really was a non-starter. And at the time we started this, we were, one of the early places to have single cell, and we had hoped to use it, but it was not going to happen. So we needed a technology that would allow us to look in a lot of places at high throughput, would have an unbiased sampling approach, and that would allow us to study both the affected and the unaffected regions separately. So long story short, we applied the first version of the technology that went on to be commercialized as Visium. We did about 1,300 mouse tissue sections, about 100 human tissue sections. And by looking at all the data together, we were able to actually notice dozens and dozens of differentially expressed genes in the ventral aspect of the spinal cord as early as we looked, which was P30, so far earlier than you're seeing in bulk measures. We took the data and standardized the expression observed in each of our spatial capture areas and clustered all of our capture areas in an unbiased way, and then looked for modules of genes that were showing correlated expression or anti-correlated expression across all of our data, then mapped that the expression of those modules back to their spatial locations. And what you can see here in the top is that our module 8, which has 1,300 genes, is a very clear progressive reactive gliosis signature, and the genes that make up this module are exactly what you would hope to see. They're all glial markers. And in module 11, it's much smaller, but they're basically all expressed in motor neurons, and you see the progressive loss in our model. So this is, this convinced us that spatial technologies were the future, and we weren't really going to deal with much else from that point forward. The problem was, at the time, the resolution was 100 microns with the 200 micron center-to-center distance. And you can see here that this, of course, immediately prompted questions, well, how do you know what cell type it's coming from still with this resolution? So long story short, we tried to develop classifiers and other automated approaches for not only counting how many cells were in each spot, but also trying to determine what they are just based on morphology from an H&E image. And that work has actually continued and has a preprint out now, but I'm not involved in that. But happily, while we were busily trying to figure out computational ways to do this, 10x took over and started really refining the spatial resolution. And in some ways, you could sort of think of this as a solved problem, especially when you consider other platforms like StereoSeq, where you actually have sub-micron scale capture features. So I won't dwell on this point because I think the main problems here now are not resolution, but in data quality and sample compatibility. So just briefly, so there's a sort of common understanding here. On the left, we see the original version of the capture strategy used in the 3' version of Visium, and on the right we see the probe-based approach. And the probe-based approach is great because even if your RNA is degraded, or more likely if you have cross-links that are preventing your reverse transcriptase from going all the way through the message, it doesn't really matter because all you have to do is land your probes in the right target, ligate them, and then also an advantage is that combined ligated probe is quite short compared to a full-length transcript. And so this probe-based approach allows you to overcome a lot of sample difficulties. The problem is that it is not truly unbiased. You do have to pre-select your target sequences and whatnot. And for some use cases, that's not really going to give you what you need. I will just briefly show you this, which is to say that this probe-based approach makes a huge difference in being able to actually obtain data from, difficult samples. And I guess, at the time we were considering FFPE difficult. But referring back to a use case in which you really don't want to pre-select the things you're looking for. In pilot work that we were doing to apply for some autism-related funding, we wanted to look at a new mouse model that was known to affect splicing, and we wanted in particular to try to identify new splicing events or cryptic splicing events. And this is obviously something you can't design a probe for without already knowing what to look for. So what we did is we took these mouse brains at a couple time points, and we isolated single nuclei from them, and we also took sections onto Visium slides, and then ran the resulting libraries on both ONT and Illumina. And it's quite nice, actually. This is just, as I said, very early data, but you can see in the leftmost panel is probably the most obvious example of this. You can see that in the upper left-hand corner of this embryonic mouse brain, that there is distinct expression patterns between this one transcript of GRIA2 versus the GRIA2 gene as revealed by Illumina sequencing. So this is really fun, and we think that this is going to be very relevant for a lot of neurodevelopmental diseases. I'll just briefly mention work by colleagues that, again, I'm not involved in, but to show the versatility of these type of approaches, you can modify the sequences on these type of slides such that they are compatible with 16S sequence capture, and you can actually see correlated expression of host genes and microbiota with spatial resolution. It's quite nice. On the left, this is in a mouse colon. On the right, this is on a leaf. So quite nice. All right, changing gears just quickly at the end. We took our data from the first work in ALS, and we found some targets we wanted to go after, but they were all clearly going to require finer resolution, and we wanted to look at things like phosphorylation and whatnot. And there wasn't really an imaging platform that would allow us to do this, and moreover, we didn't have a CapEx budget. And so we landed on a technique called 4i that lets you use any antibody you want to iteratively stain your tissue and build up a high-plex readout. And this is the first experiment we did with this, and it's always great when you try a new technique and it just works. So Joanna just immediately was able to get, I think it was like a 15-plex experiment. I'm showing you just some of the data here. And so we knew we wanted to automate this because this was done by hand. So we, again, didn't have CapEx, so we started trying to figure out how we would do this. Standard imaging flow cells are quite expensive, and so is all the stage adapters and all this that you need. So we couldn't do that. So we developed a technique using a Cricut, which is an instrument designed for cutting greeting cards, to cut this capped-on tape, which we used to make a sort of gasket between a slide and a cover glass. And we drill a couple holes. In this version, we also attached a couple ports with just PDMS. But in a serendipitous bit of timing, right when we started doing this, Illumina announced the end-of-life roadmap for their HiSeq family instruments. And so we all of a sudden had over a dozen of these things to play with. So we started tearing them down and figuring out how to control every component inside. We modified our flow cell so it's adaptable, it's compatible with the Illumina stage. And these things are great. I mean, internally it's quite similar to any of the state-of-the-art boxes in terms of the optics. The detectors aren't as good, but it's still quite good. You have temperature control, large number of refrigerated reagent reservoirs and whatnot. And we were able to automate this 4i workflow. So I'm showing you 14 channels here on the right of a 21-channel experiment. And on the left, I'm showing you k-means clustering of the various protein expression patterns here. So as you can see, I mean, the images are they're pretty nice for a machine that you can get for the cost of shipping. One problem with 4i, though, is that it does still depend on indirect immunofluorescence, and so species incompatibility is an issue. And the assay time, as a result, can be a week or something. So we wanted a much faster approach. So we set about developing a PySeq workflow based on Immunosaber, in which you put all of the antibodies on at once, and they are oligo-labeled, and you can do cyclic imaging and elution and staining, and very rapidly build up a nice high-plex protein-level image. We've done 40 proteins on it, and I'm showing you on the lower right here a nice little surprise that we had, which was that we inadvertently targeted the line 1 element by selecting the wrong reference genome when designing probes where repeats were masked. So we ended up realizing that we had a probe against this transposable element, and it may be difficult for you to see, but in purple we can see that the DNA level presence of that in nuclei, and in green protein level expression of GFAP. And just briefly, we're now using the PySeq to do imaging-based spatial transcriptomics, and this is based on StarMap. So it's quite nice. It's cleared tissue, rolling circle amplification, and during RCA, you actually covalently link the Rollini into the gel, and so it's super stable, and you can image this thing through dozens of cycles, throw it in the fridge for a couple months, pull it out and image it again, and it'll look great. So just to conclude things really quickly, you really need to think about the questions that you're trying to ask before you select your technology. We've been very fortunate in that we've also had an opportunity to engage our collaborators before samples were collected, and so we can help design the way that samples are prepared that is good for this. But the reality is there's not one platform that's going to be sort of the jack of all trades and work in every scenario. And so it's essential that there's a close collaboration between the spatial core and the end user with the samples, to make sure that everybody's on the same page about what you're trying to get out of the work. And just briefly, I'll plug our PySeq platform. We're very proud of it and excited about it, and yeah, we'd love to engage with anybody interested in running projects or in dusting off one of their old retired sequencers and converting it themselves. So with that, I'll say thank you and head off to Ioannis.
All right, good morning from me as well. Thank you all for joining this session. Before I begin, I wanted to thank the organizers and our sponsor for bringing the community together. ABRF is a unique, I think, conference just because it brings core facilities. And I'm leaving Vegas with a lot of ideas, new collaborations, and a stronger sense of belonging. And I think I really, really needed that. So yeah, I think my colleagues teed it up perfectly for me. I'm focusing on the navigating portion. And just a little bit about me, I'm faculty at Harvard Medical School. I'm running a lab at Beth Israel Deaconess Medical Center and 2 core facilities. One of the facilities is the Spatial Technologies Unit, which will be quite a part of the focus. So it was established back in 2021 as a freestanding center for single-cell and spatial service provision, about one-to-one revenue from internal and external clients. So we work with cores across Massachusetts and across the US. So we work with biotech, pharma. We're super collaborative and happy to work with you. And also it was with a lot of support from the state of Massachusetts. So back in 2020, when we were designing the facility, these were the first pillars we identified. We wanted to provide access to technology, to support training and education to everybody, researchers, students, but also physicians, investors, venture capital for this new field, support IP generation, and bridge the way for these technologies to reach clinical care. I'm faculty in the Department of Pathology, so that was a personal goal. The whole spiel today will be material access technology. And should I press again? Okay, so this is roughly our ecosystem. We try to support our community across the resolution axis. I'll just briefly mention the assays. Technologies change so quickly, so it's not so specific for today. GeoMx from Brugger, Visium, we do direct placement, version 2, HT, protein side, PhenoImager HT, and phencycler fusion from Akoya, and this imaging-based spatial transcriptomics. Xenium, we have G4X now in production line. It was early access last year. Now it is in production line, Merscope, and the Voxel for SuperRes. What we definitely take pride into is our pipeline. So we are a completely vertical unit. We can start from processing the tissue, create microarrays, we section, we can create single cell data from the same samples, even if it's FFPE. A lot of investment in robotics and automation to make the assays production grade. We sequence in-house, we analyze the data in-house, so a client can come in with a tissue block and a question, and they can leave with figure 1, 2, 3 if that's what they want to. Maybe it's me. Okay, so this slide that happened during Vegas, so many colleagues asked me, can you provide a spoiler alert for the G4X? This will be in a future talk, but this is our experience for now. These are 3 instruments. They're in our spatial room for early access, and it has great prudential center view. And this is how the actual interface looks like. You have 4 flow cells. They're independent. You have 10 square centimeters per flow cell, and each flow cell has 4 lanes, and you can put a different probe set per lane. So you can have 16 different probe sets. You have different size conformations, so you can do, there's a flow cell with 32 samples, and there's a flow cell with half that. So if you have 4 flow cells with 32 samples, that's 128 in the big run. And we tried initially to try to give it simple stuff, breast cancer, but it worked really well in our hands. We haven't lost a sample, so we went up skin, bone marrow, vessels, the worst things we could throw at it. I mean, it works normally. This is a marrow, for instance, example, human, archival, horrible sample, and it works quite well. We worked with Singular in 3 different versions of the chemistry, and there was dramatic improvement as these chemistries became in the protocol more mature. But we always took serial sections. We had some samples we did every single run, and we see serial sections within the same run. Red is the negative probes, and blue are the actual targets, and this is the same serial sections between runs with 0.99 correlation. So going back to the topic of navigation, core facilities make assay selections every single day. We choose assays to, are we going to bring this assay to our facility? Are we going to do early access? Or is this the right assay for this project that I'm discussing with my client every single day? That's what we do. So one of the most difficult decisions is which assay to invest in. It's not a simple decision, it's a business decision, it's a community decision that we're supporting, and it has a mix of both and can be different at different times and different cores. So we focus, I'll try to walk you through what is going on in our brain, and maybe that's useful. What makes us a very specific case, and might not apply to you, is that we're not subsidized at all. So we're cost recovery center at the 100% rate, so that makes our decisions a very business-focused decision. First of all, we try to capture every cost for a new assay, and that's not easy. Even for us, we've done it so many times because you have to calculate CapEx. If it needs be supporting equipment, sometimes it's not easy to find exactly what you need. What would it mean for my facility if I have this new assay? What do I need to change my pipeline to support it? That's an additional cost. It's time, it's FTE, it's thought, it's effort. If I want to automate it, how much would it cost? If I want to bring it into production, this means SOPing and training, and means test runs. These also cost, and it's FTE. FTE to establish, FTE to run outreach activities. How will the community know I have this? I will be doing workshops, I will be doing talks, I will be doing seminars. This is time for my people not being on the service line. What's the service cost? What's the data analysis cost? How is data storage handled? And then the need, which is the good stuff. Is this a current need? Is this a future need? Does it give me a strategic advantage? Does it support new grants? Is it my ecosystem or everybody needs it? And if I don't have it, what can my users do? Can they go somewhere else or nowhere? And then this is a return of investment. That's complicated. How much time do I need to roll out? Do I have clients waiting? What is the throughput of the instrument? How many assays can I push per week in realistic terms with the FTE that I have? What is the lifetime of this instrument? Should I recover in 5 years or should I recover in 2 years? And then you have to take all these into account to make your decision. And everybody thinks, so I cannot go back and forth very easily. Okay, I can. So everybody thinks this when they're getting an instrument, but I think this is very important as well. Have you seen it on the bench? Do you know how it is in your hands, in your environment? How is the prep? So early access is super essential. Have you seen real data? Do you have anyone that has real data? What is their experience? Has it been benchmarked? How quickly can I adjust to what this instrument needs? Do I have users with samples ready to go, or do I need to start looking for users after it's in? Can I handle the data? Can I analyze it? Does my bioinformatics support? Can do that? All those things can make or break you, and you can lose 6 months. And in an instrument that has 3 years, for instance, lifetime, 6 months would be a significant hit for your ROI. And what's your competitive space? So early access, I mentioned this word multiple times, so a slide had to appear. Early access, sometimes you think, oh, I'll get a free sample, or I'll get the free kit. There's nothing free. And often, vendors forget about this, and often we forget about it because we're super excited. If I send the free, if I send some samples and I get data back, the computational biologists will have to stop what they're doing supporting clients and work on that data. We'll have to sit down and discuss. People will have to think about it. If they send us a kit or an instrument, wet lab FTE, my FTE, my team's FTE, space, networking, data storage, computational, this is cost. So of course there's this business side for early access that you have to identify, but also it's the opportunity. If this thing works, what does it mean to the community? If this is a good assay, what will be the impact? What needs does it cover? What's the timeline? And also, how open is the company to hear my opinion? If I raise the alarm, ring the alarm, like I don't like this in this assay, like the bench work or something, will they listen to me, or this is early access to just check the box that they worked with our facility, and that's it. And of course, this is difficult. Which assay is the right, for this project? And you talk with the PI, and sometimes there's a lot of confusion what they want to do, or sometimes even worse, they have a very specific idea what they want to do, and it's not the right approach for their problem. So we focus a lot on the second book of Sun Tzu, which is The Art of the Core Consultation. Didn't sell as much as the first one. So the first one, before you begin talking to a client, you have to agree internally. What is your core strategy? Different cores have different needs. So for instance, can I do the things that Silas is doing? Do I have the bandwidth to do custom assays if need be? Up to that point, how much risk I can take? Do I like smaller projects or do I like bigger projects? Do I like projects that are cheap or do I like projects that are very long and very expensive? Does the income that I will be getting affect my decision? If this project, covers 2 people for 2 years, will change my decision versus, so first of all, you have to identify what is important for you. For us, we're very clear, and it's not very common, but I think it works really well for us. Project success is what the PI thinks is going to be successful. And sometimes this hurts us, time-wise, financially, it's not a good short-term decision, but long-term, I think it pays off manifold. And how do you achieve this? Everybody has to go through a 1-hour consultation if it's a new project. Even if you have worked with them 1,000 times, even this is the most experienced group, every new project, 1-hour consultation with 4 or 5 people on the call, director level, computational biologists, everybody on the call, every single time. like no questions asked. Then the first phase of the consultation is, what is success? So first we talk science. No instruments, no spatial, the words should not even be mentioned. First we talk about the project, the research questions, primary, secondary, and then when we feel we understand the context, we ask directly, what is success to you? Talk about the manuscript, talk about the grant. Like, just talk about it and dream, what would be the best outcome for you. And then we go to the logistics phase. Samples, dimensions, data. Do you have already data? Single cell, bulk, budget, timelines. Do you have hard deadlines, soft deadlines? Can you get the data analyzed if we give them to you, or should we plan for the analysis as well in our timelines? And then when can you actually start? That is very tricky to define because there are many traps, in this discussion. Are there MTAs that you need to put in place with somebody to give you the samples? Do you have the samples? Can you share the samples? Does your IRB approve you to doing things with the samples? Are you waiting for a milestone? Are you waiting for funding? Are you waiting for anything? And sometimes they do, and you need to find that. And the phase 3 is discuss options. So where you're having this discussion, this is a toy example. I added the instruments that we have. I added also the CosMx from Brittany because we don't have it, but I wanted you to see it. Of course, this is fake as an example. It's very simple. So you're having this consultation, and they say, I want to do 10 samples, FFPE, it's big, and I want to focus on rare cell population. So in your head, you immediately think about, okay, I have this assay and this assay. And then they tell you, no, it's actually a different project, 500 samples, an FFPE and human, and I want to do 3 months. So then it's very easy for you. You have a single option. But sometimes it's not as easy. It's more nuanced. They tell you, I want to do FFPE human, and I want to do pathway analysis in situ. When we hear that, it's usually whole transcriptome. But nowadays, you also have instruments that are imaging-based that do 5,000 genes and 6,000 genes. So you have to weigh that option. It's not all pathways, but a lot of pathways and have different. So that's why I have marked them there. So that needs more thought, and it's more nuanced, and you need to discuss more than that. Or for instance, this one, you might say, this is pathway analysis in situ, but I like cell-cell communication. So budget-wise, the V2 might be great for you, but also this, you have to explain things. So you reach a point in your consultation. Oh, and I wanted to mention the consultation gap. So sometimes you don't have a solution for what the client just said. And that's an important one, so you have to take this into account. So in the final phase, you juxtapose the available options and you explain differences in cost, timelines, success. You discuss your previous experience very openly. I have worked with this tissue in this instrument, which seems the right one, but you have higher risk versus this assay, which might not be the perfect for you, but it's safer. You have to be very clear. You have to make direct recommendations, which is something many, even for my team, they don't want to say directly, I think you have more success with this, but we know the instruments better than anyone. So we have to speak very, very openly. And sometimes we do the hat switch, which is the PI hat, and we provide them insight information, if you want this for a manuscript and I saw this as a reviewer, I would have raised this, and that, so you should have this already ready. Or for an NIH reviewer, or as a PI, sometimes I want to do more with the money I have. So maybe you don't do that many samples, and you do single cell, and you do this, and you do that. So sometimes you give practical advice. And the consultation gap I mentioned previously, it creates a virtuous circle because you identify a gap, and if that gap happens every time or very often, then you might need a new assay. To bring it into your community. There is demand. But maybe sometimes this assay doesn't exist, and you have to reach out to the vendor and say, I get a lot of consultations for Visium and microbiome or something. And you don't have that. Maybe we do something about it. And then you might do an early access. And if you do the early access and you like it, you add this to your arsenal. But then if you start hitting new gaps after 5-6 months, then there is this new demand that you need to cover, and this is the virtuous cycle. Major hurdles, there are no commonly accepted standards. There is no common accepted metrics. You see FDR in one instrument and FDR in the other instrument, but the formula underneath is different, even though the word is the same. There are different, you see a benchmarking paper and another benchmarking paper, but the metrics are different. So it's very difficult, even for us, for the community to decide. No core has experience for everything. Even if we do a lot of different tissues and a lot of different species, it's very difficult. And early access, sometimes you raise the alarm. Like there is this issue with this assay, and I think before you have it commercial, you should do this. And sometimes, there's nobody there. Sometimes everything changes and it's awesome. So what do I do? First of all, I love this, that it exists. The WhatsApp from ABRF is, we ask questions all the time, we get answers all the time, and we try to answer as much as we can. But if you want to reach also to the labs, not the cores, like, would you use this? Would you be interested in this or whatever? You can reach out to the labs through Gestalt. There is a WhatsApp there as well. I wanted to thank the facility, my lab, our funders that make everything possible. We're super collaborative. Find me afterwards or reach out. Happy to take any questions. Thank you.
Okay, if anyone has questions, you can line up at either one of the two microphones and we can discuss.
Really a great set of presentations. So I have two questions. One is very amazing work in repurposing a HiSeq. It reminds me when they went from the slab gel 377 to the capillary instruments, we repurposed the 377 for protein gels. Basically that's the same concept of what you've done. What I'm wondering is, do you think the work that you've done with the HiSeq could also be applied to an old NextSeq 6000 to repurpose those platforms as well?
Are these working? I don't think. Yeah. Can we turn these on or? No? Okay, there we go. Internally they're a little different. The HiSeqs used 4-color chemistry, which definitely goes a long way for doing higher, like fewer cycles to get to the same number of measurements. We haven't played around with them, so I suppose it's probably possible. I will say, though, that there is some variety in Illumina's hardware, and the HiSeq family is really amenable to hacking. It's very easy to basically get at anything through simple serial commands in Python. But the CBot, which is the companion instrument, runs an embedded version of Windows, and it's something that's much more difficult for us to actually, muck around with, even though that is an instrument we'd love to have for some other things. So I'm sorry I don't have a clear answer on that exact instrument, but it's just because we haven't tried. It may be possible.
Yeah, I just Googled because I don't remember the name of the assay. There was a group that repurposed NovaSeq 6000 for Spatial maybe a few years ago. But I don't remember, there are like many acronyms and this one. But if, happy to share the manuscript, but a team did that. And if they did it for Spatial, I'm pretty sure they figured out a way to control the microfluidics.
Maybe we can talk offline. I'd be happy to, help pull together a group that might want to do that, since we have a 6000, that's just sitting in a corner.
Yeah, we actually have one partially torn down right now. We're actually working on sorry, I thought you said a NextSeq, but the NovaSeq, we have one in the process of being dissected. So be happy to, to discuss it offline. Thanks.
Okay, that's great. So my second question relates to the last speaker was talking about the fact that there are no standards, there's no, comparable metrics, even though some groups have tried to do that internally within the group of instruments they've had. The group from the Broad showed something at the AGBT meeting just for the instruments that they had in-house. A number of years ago, as you well know, there was a suggestion for the ABRF groups to do equivalent to what we did for NGS, which was to compare all the commercially available spatial multiomics platforms. And that was a little bit too early in their development. They were changing too rapidly. I've had some discussions with folks, so it seems it might be the right time to do that. You know, I've talked to some really good colleagues as well as folks at NIST that are interested in supporting that type of moving forward. And I'm just wondering about your opinion. Is this a good time? You know, is this actually a feasible type of project to move forward for the really great research groups at the ABRF?
Yeah, so the answer is yes. The main reason, I just mentioned briefly Gestalt, the main reason why Gestalt exists is because of that. You know, we were like 10 friends and we identified that, you know, we cannot even talk to each other. So that's why we thought we would be 10 people just coordinating, and currently after a year, it's like 1,500 people from 45 countries. And one major initiative, which I'm co-chairing, is the Standards Working Group, and we're talking with NIST. And I think as soon as there is a plan, the next location we will involve is a GRG group from ABRF, because at the end of the day, it's the core facilities that do all these things. So if ABRF is massive, you know, it's an incredible partner. So if we're able to put together, what we don't want to do is what we have seen in other fields where 5 standards show up, and that's even worse than no standards. So if we arrange for the whole community to talk to each other and reach an alignment before something is announced, I think that will also be successful, because then we will also be the people that will be adopting it. And if we already like it, we will adopt it, you know, as it is. So yes, that's why I had also the slide. I think it's high time. We usually fields start from Wild West. They go to an organic growth, they identify the need, and there is a meandering path how to cover that need. Spatial has reached the point where that need is identified, but if we talk to each other, we would not have to try again. We can just cut directly, and we've done this 50 times as a field, macros, bulk, single cell, exomes. Why can't we just do it the easy way this time?
No, I think that'd be great. You know, I'm happy to help facilitate getting people together, hopefully even today before the end of the meeting, but if not, you know, sometime soon. So anybody that's interested in that type of large-scale, very typical ABRF project, you know,
Let's
please
go.
let me know. Yeah, thanks.
I'll just,
Oh, and by the way, for networking purposes, I forgot to start off with, my name's George Grills. I'm at the University of Miami. I'm sorry.
Nice to meet you.
I was just going to say thank you to Ioannis and the Gestalt people, because I think this is the only way this is going to be feasible. If we really want to benchmark things, we're going to need standard samples, which is going to mean MTAs and things like this. And if we don't have a centralized way to coordinate that, it just won't happen. It'll be a mess. So thank you.
Thanks.
Really great session. Amanda Caney from Fred Hutch Cancer Center. Ioannis, I was really happy to hear the emphasis on that initial consultation, that we found that to be super important, getting everybody in the room and all the expertise in the room to help. Some of these projects can be quite complicated, and that consultation doesn't end right there. It goes back and forth over a period of months oftentimes. Do you try in any way to recover those costs, and how do you go about something like that?
That's a great question. And so what we have decided to do, which again is not perfect, and again we're not subsidized, so we're very, very pushed to identify all our costs, and this is another cost. So we do 2 things. Number one is for projects that are very complicated, we're the only HabMap tissue mapping center in Massachusetts, which is great. It sounds wonderful. I use it on my slides and my grant applications, but that means a lot of overhead. It's not that if I take 5 samples and do this, or we have, as a PI, I have a PO1 that's really stressing the facility as a client. Or there are RO1s that have 55 degrees of freedom. So we usually tell them, why don't you support a 10% or 20% FTE on that project to act as a manager, you know, for this. And we find the right person, and that person will be a contact. That has a 15 to 20% success rate. Not all groups are like interested in doing that. The other thing we do is we identify which assays usually have a lot of overhead. Let's say custom Xenium. Custom Xenium is never the cost of, the probe set. So we bake in 5 hours or 10 hours of consultation during that, and if we exceed that, we cost per hour. But we have a portion of that, the average time or the minimum time, we have that baked in. Because it will be there. It's always there. There's no way it won't be there. Yeah, so that's it. I have no easy answer. But what we do is we try to bake in a portion of the cost, usually the minimum, and then we have a per hour consultation rate, and we start charging after that.
No, that's really helpful to hear. Just curious if you've got pathology support as part of that team, and does that factor in?
Yeah, so I didn't go there as a member of the Department of Pathology as faculty. This gives me really easy access to faculty. And we have done this in the past. So many of these assays, for instance, the assay that Meteor showed, when I saw this need the pathologist before, or GeoMx that was presented previously. The success of these assays is pathology consultation before. So we have the ability we have established it took a while, to engage pathology support and also to bake in costs for their time as consultation time, as they're paid reviewing clinical cases, we have the same rate when they're, of course, they also part scientifically or anything, but we have cost to recover their time when they do that. Yes, I raised this to NanoString before it became broker, that some of the GeoMx website ads were misleading because there were just random circles on a slide, and that's not how you do this assay. So yes, pathology 100% before, and pathology 100% after. Now, with all these AI opportunities, we'll see how that play out, but for now, it's human in the beginning, human in the middle, human in the end, for sure.
Thank you.
On that point, we roll in both a dedicated project manager and a computational biology component in our costs from the beginning, and we have internal conversations, independent of our conversations with the collaborator. One pain point for us is that as an independent genomics institute, we don't have pathologists, and that is always one of the things, if we're working on a new tissue, we don't know what it ought to look like, and we're the ones doing the sectioning. It's really hard to know if we're looking at the right part of the tissue and all this, so we always have to do extra lab work there, and it would be nice if we could put that as part of the whole package of consultation.
On that front, would it be helpful if there was a group of pathologists that were sort of identified for this type of work?
Yes. Okay. Where do I buy this?
Yeah. I've been hearing this a lot. I am a research pathologist, and I've been hearing this a lot during this meeting for different projects, and we're thinking about putting a session together for next year, perhaps. But anyway, this has got me thinking. Thank you.
Yeah, we need it.
100%.
Yeah.
Hey, Sean Williams, University of Miami. We've also found that sort of three-step consultation upstream to be very useful. And just a sort of operational question, what platforms do you use to record and document your consultations? And do you use platforms where users can update things offline or after a meeting, or do you sort of lock down at the end of each consult?
So that's complicated. As an answer, for now, we're using artificial intelligence, which means we have a great program manager, and that program manager distills everything. We thought to get transcripts of the calls or recorded, but at the end of the day, somebody has to make sense out of all of this and provide a distillation of the consultation in bullet points as an email to the client. So even if we discuss 55,000 things, at the end of the day is what they will agree to that email. So we haven't found an easy way to do it, and we don't want them to be updating anything that doesn't go through email. we were initially very flexible, and we've had like key personnel like come to our facility and tell us, you know, we decided to double the number of samples or something like that. And we were very happy, but when they got the bill and the, you know, the person paying was not the person asking double the number of samples and was dozens of thousands of dollars difference, that created issues for us. So no, not even iLabs. There has to be an email with everybody CC that uses plain English and says, yes, I approve this change or this addition. We have found humans to be the weakest part of that interaction.
Thanks.
Yeah, I mean, if there is a good tool, we would adopt this 110% as addition to our CRM.
Yeah, no, we struggle with that as well. That's why I asked the question.
Yeah, we also just use email chains usually.
So I have a question regarding the data archiving. So all these platforms spits out a large number of files, different formats. So is there any guidance, like, are you archiving all files, or you're just selecting something? What are you ready to lose? What are your thoughts on that?
That's a very sore subject for us. I will say, so I have a CosMx, and you can imagine the data that comes off of that. My strategy is to push all the data to the researcher, because I don't have enough budget nor space to store all the data that I'm getting. So once I get the data back, I'll do whatever like conversions I need to do, and then I'm handing that right off to the researcher and telling them that's their responsibility now. I'll keep it for about a month just to make sure everything's fine, but after that, I need the space for somebody else's project at that point.
Is that actually what NIH mandated, that we need to keep data? Like as a core facility, we need to archive
Not
them?
the core. So not the core. That's the PI's responsibility for the.
Okay.
Yep.
But the reality of it is, how much do you trust your clients that they will keep the data?
Yep.
As a client, I have reached out to core facilities in the past multiple times, you know, my dog ate my homework kind of question. There was a postdoc that left and deleted their whatever and deleted everything. Do you still have it? So this is its reality, and it happens. And for spatial and single cell, and when the data sets are terabytes, how much do you trust them? Where are they going to keep it? You know, so we offer data storage as a service. We're really good at having a very cost-effective cloud footprint, and happy to discuss what cost-effective means, but it's quite good. We have our own iron, like local store, and we provide it as a service. And in our TNC, we tell them like all data will be purged after 6 months, but we don't. And eventually, when our capital for these things reaches its limit, we go like 2-3 years in the past and we start deleting. But our TNC says your data will disappear after 6 months. So, and we give them an email like your data will disappear then, but we don't because we don't trust them.
It is an interesting question, though. I mean, you know, computational techniques are developing really quickly, and it's clear that if you had the raw data, there's probably going to be a better way to look at it in a couple of years. But the cost of this scale of data storage is prohibitive for that. And I think it is an important issue that I don't think we've actually really confronted as a field yet, you know.
Yeah, and if you're a cost recovery center, like if you don't have any additional like profit, if you get your cloud store very cheaply, you can offer cloud store or local store or something store very cheaply to the PIs, and it wouldn't make sense for them to just end their problem with a few $100s per year, and they don't even have to think about storing long-term and meeting NIH regulations and things like that. You just make that problem disappear for them. So it is a service we provide. It's not something we're like super happy about, but if we don't, then who?
Thank you.
All right, I think we have time
Sorry.
for one more. Yep.
Thanks. It's not actually a question, it's a plug for a session coming up right after lunch, research data management strategies for cores.
Yes, we'll all be
What
there.
was this arranged?
Thank you. Perfect.
This was planned, wasn't
yeah,
it?
that's in bronze 4, 130. So come with questions. Thanks.
Of course.
All right, thank you guys for coming to the session.
Thank you.